Hello everyone. So in this tutorial we will learn about how RBC is synthesized in our body. First of all, what erythropoiesis means. It is the process of synthesizing erythrocytes, and like erythro means red blood cell, that is erythrocyte, and poiesis means synthesis. So synthesis, the process of synthesis of red blood cell is known as the erythropoiesis. Now there are different sites in our life. From the beginning of our life to end, we have different stages and different phases of synthesis and production of RBC. So let us see from the first. From the 2 months of intrauterine life, we have got the mesoblastic stage in which yolk sac produces the RBC. And then we have, after that 2 month or 3 to 6th month of the intrauterine life, we have hepatic stage in which liver and spleen secretes or synthesize the RBC. Similarly, we have in the last 3 months of pregnancy, or let us say about the 6 to 9 months of the pregnancy, the fetus is developed intrauterine, and that their bone marrow and liver is like producing the RBC. So this is the intrauterine life. After the birth, usually in less than 20 years of our age, long bone and all flat bones synthesize the red blood cells. Whereas after the 20 years of the age, membranous bones, which are the sternum or skull or vertebrae, that might the marrow, that is these bones will secrete or synthesize the RBC in our body. So this is the different sites at different ages in our body. Now let us see about the factors. What are the factors required and how they help in the production of RBC. Talking about the factor, we can classify them into general factor, maturation factors, and factors necessary for hemoglobin synthesis. So let us see about the general factors. First of all, we have the erythropoietin. This is the overall stimulation of the bone marrow. It does the stimulation of bone marrow. It stimulates the bone marrow, which will increase the secretion of hematopoietic stem cell, and in that way there will be the high or increased secretion of RBC. We have thyroxine, which will increase our basal metabolic rate, we have studied, but instead of that, it also boosts different functions in our body, different cellular functions gene production. So in that way it will help in the production of RBC. Similarly, there are different vitamins such as B, C, D, E. It will all help in the synthesis of RBC. Whereas we have growth factors such as interleukin 3, 6 and 11. These all growth factors help in production and synthesis of RBC. And erythropoietin is one of the important factor which is further regulating the release and production of RBC in our life. So erythropoietin is induced in hypoxic condition, where it will be secreted by the juxtaglomerular cells of the kidney, and then that will stimulate the bone marrow and production of RBC increases in any condition required. Then after we have the maturation factor. Maturation factor means now the cells, their cells are now in their immature state. Now it has to develop into a matured state of RBC. So these factors are vitamin B12, folic acid and intrinsic factor. These all help in DNA synthesis and maturation of RBC. So, and this intrinsic factor helps in absorption of this vitamin B12, which is secreted by the parietal cells of the gastric gland, in our stomach. So in the deficiency of this intrinsic factor, we can get the pernicious anemia, that is slow and subtle but risky one. Anemia is risky, pernicious anemia. Whereas in the deficiency of folic acid, we can get the megaloblastic anemia, and that might be caused, there are several causes, but major one is this is the nutritional deficiency might cause megaloblastic as well as this pernicious anemia. So now likewise, let's talk about the factors necessary for hemoglobin synthesis. As we know, hemoglobin is synthesized with the help of iron, for the heme part of the, and globin is the another part. So globin is the protein part. See, this is protein and amino acids are also required. So globin is the protein part, whereas heme is formed by this iron. And this is the carrier of oxygen in our hemoglobin actually. So it is required. So iron. iron. Then after we have proteins and amino acid. Majorly we have understood about the globin. Then after we have globin consists of this alpha beta chain, and we will talk separately in our different topic about the hemoglobin. Then we need the cobalt and nickel as well. Similarly, we need like vitamin C, nicotinic acid, riboflavin, that is vitamin B2, and pyridoxine, vitamin B6 as well for the synthesis of RBC, that is erythrocyte. So these are all the factors which are very much important to learn and understand. Now let us see the different stages of erythropoiesis, how erythropoiesis occurs now. So talking about the stages of erythropoiesis, we have got, first of all, we have the primary hematopoietic stem cells, which are like, first of all, uncommitted. Then they get developed into the committed primary, that is pluripotent hematopoietic stem cells, right? So if they are like now first of all stem cell, then it will be like converted into the committed primary, that is pluripotent hematopoietic stem cells. Pluripotent means it can go to the develop the multiple cell lineages in our body. So then after this committed pluripotent hematopoietic stem cell will get converted into the colony forming blastocyte. These are all the cell lineages, like and then it will be also like differentiating into the lymphoid stem cell, and this will give the lymphocytes. And then we have got the that is colony forming blastocyte, which will further give colony forming unit for erythrocyte. Then we will also have the colony forming unit of this megakaryocyte, which will later on develop into the platelets. And then we have the colony forming unit of granulocyte monocyte, which means it will give the granulocyte as well as monocyte. Monocyte later will be converted into the macrophages, whereas in granulocyte we will get the eosinophil, basophils and neutrophils, right? Then erythrocyte, now after this stage, after this commitment, now it is like committed, and now there will be the growth factors would be achieving like coming here and reaching here, that will stimulate different stages, and these stages are here in this slide. So let's talk about the important thing that happened in the stages. So first of all, we have the proerythroblast. Proerythroblast is a very like basophilic stage. See, bluish in color, the cytoplasm is bluish, and then it has high nuclear to cytoplasmic ratio, and there is presence of chromatin and few nucleoli, and nucleoli present there disappears in this stage. This is early normoblast, and chromatin is condensed in this stage. Chromatin is condensed in this stage. Now we have the intermediate normoblast, which is polychromatic, where the hemoglobin synthesis, as though it has like started from this stage, but it gets accumulated mostly in this one. So hemoglobin, so turning it into the polychromatic stage. Then after we have got like late normoblast. Now let us see proerythroblast, early normoblast, intermediate normoblast and late normoblast. Proerythroblast, normoblast, which is early, intermediate and late, and then we have got the reticulocyte and mature RBC, and all these take of about 7 days. So maturation occurs for 2 days, whereas these all stages goes on for 5 days, and these all up to these stages the RBC has the potential of dividing mitotically, but after this stage they loses the cell dividing capacity. Later on it will have no nucleus, no any organelles and at this stage reticulocyte, the organelles and the nucleus of chromatin are dispersed and rupture, and that under microscope appears as a reticular or reticular network that gives us and thus we call it a reticulocyte stage. Then after we have got the mature RBC, which have its own central pallor, and this is the surrounding biconcave shape. So this is all about the erythropoiesis. Now let me talk very slightly about the important events in these stages. So synthesis of hemoglobin starts in proerythroblast. Similarly, nucleolus disappear in this early normoblast. In intermediate normoblast, hemoglobin starts appearing. Hemoglobin appears in this intermediate normoblast. Similarly, we have the late normoblast, in which nucleus disappears. Nucleus disappears in late normoblast. And we have the reticulocytes, where we have studied the reticulum is formed, and then cell enters the capillary and to the site of production, from the site of production to the capillary. Then in this stage the reticulum disappears and attains the biconcavity. So this is about the maturation. So this is all about the synthesis, production, and the factors and site of the RBC. So do comment below for any queries. See you again. Bye bye.